Okay, so today we're going to be talking about transcription. And transcription is a topic that is pretty complex, but for the case of what we need to know and what we're going to be asked on board exams and stuff like that, it's all very general. There is some detail, but it's nothing that you really haven't seen before. So, my goals for this video are to cover these 4 things. So, we should be able to list the different classes of RNAs produced from the genome and describe the function for those RNAs. We should also be able to reproduce the stages of transcription and know the differences between eukaryotic and prokaryotic transcription. We should also be able to draw a cartoon of a transcriptional unit and understand the role of transcription factors in contributing to the complexity of gene expression. And finally, we should be able to discuss the basis for epigenetics and how modifications of both the chromatin and DNA affect rates and levels of transcription. Beyond this, we're also going to talk about the Lac Operon at the very end and how that's a method of transcriptional regulation seen within prokaryotes. So let's get started with the different types of RNA. So there's 3 main types of RNA that we discussed. The professor went into a little bit more detail on some of the smaller ones, but for the sake of the video, I'm going to leave that out. The 3 main ones are, I'm sure almost everybody has heard of these: mRNA, tRNA, and rRNA. Okay. And if you don't remember, mRNA is essentially what's produced by RNA polymerase 2 during transcription. So when we transcribe something, we take our DNA, RNA polymerase 2 comes in, transcribes a gene, and creates a messenger RNA, what is known as this mRNA. And this can later on go out into the ribosome to be translated. Okay, mRNA goes out and gets translated. That is our messenger RNA. That is what encodes a gene product that we're going to later turn into protein. tRNAs are made by RNA polymerase 3. And what tRNAs do is they're the RNAs that carry the amino acids to the ribosome. And if you remember, tRNAs have that kind of weird, this is a terrible drawing, but for the sake of whatever, they have that weird function, or shape, sorry. Finally, we have rRNAs, and rRNAs are produced by RNA polymerase 1, and rRNA is largely what makes up the ribosome. So that's the ribosomal RNA. And the ribosomal RNA combines with proteins to form the ribosome, which is one of the key players in translation, basically translating this mRNA message into a functional protein. So now that we've talked about the types of RNA, we're going to kind of get into the different stages of transcription and what these stages do each and what the transcriptional units and things like that look like. So the first step of transcription is what we call initiation. And just like everything else, our body's really good at regulating when we want to produce certain genes and when we don't. So, in order to produce certain genes through transcription, we need a few things. We need these guys. And what these guys are their transcription factors. Transcription factors. And essentially, they're just proteins that have to exist in the nucleus to recruit and help RNA polymerase find the promoter region or the promoter sequence within a given set of genes that we want to transcribe. Okay? So once all of these transcription factors are present and we have the machinery necessary to begin transcription, RNA polymerase can then bind to different promoter regions here. This is the TATA box. This is a site where RNA polymerase can bind. We also have the CAT box, which is another site where RNA polymerase can bind. And RNA polymerase will bind, and it'll run along this strand. It'll run along that strand of DNA and essentially transcribe it into mRNA, or pre-mRNA. Okay? And that is the elongation step. Again, the elongation step is once our RNA polymerase has been recruited to the promoter region, it runs along the DNA and it spits out a pre-messenger RNA. Basically spits out a transcript that we're going to later turn into protein. And finally, we have a termination step. And this termination step, one key point, is that termination usually occurs in an area of the DNA that is rich in GC content. And why is that? You might be asking why that is. If you remember, A and T nucleotides, when they base pair, they only have 2 hydrogen bonds. So their melting point and the ability to separate those strands is pretty easy comparatively to the GC rich content areas on DNA. Those have 3 hydrogen bonds. So when we have our DNA or our RNA polymerase running into an area where it's harder to break these hydrogen bonds, it's actually going to signal it to stop. So those are essentially the 3 different stages of transcription. Next, we're going to move into the major differences between prokaryotic and eukaryotic transcription. And there's 5 main differences that I want to kind of talk about. So the first main difference is that in prokaryotes, RNA polymerase can directly recognize sequences in the DNA and bind to it. It does not need a promoter region. Okay? So in eukaryotes, we do not need a promoter region. Okay? Number 2 is that prokaryotes have no nuclear envelope. There's no compartmentalization of transcription and translation. In eukaryotes, we have to transcribe our DNA to RNA in the nucleus and then ship it out into the cytoplasm where it's going to get translated by the ribosome. In prokaryotes, everything happens simultaneously. So we have no compartmentalization. Essentially, prokaryotes have no nucleus, so transcription and translation can happen simultaneously. The third difference is that prokaryotes don't have introns. Okay? There's a lot of post-transcriptional modifications that happen to our mRNA, which we're going to talk about in a little bit. Actually, we're not going to talk about those. But the 2 main mechanisms are splicing out of introns and essentially adding a 5' cap and a poly A tail to it. In prokaryotes, we don't need to do that. There are no introns, so we don't have to process our RNA once it's been made. The fourth difference, in which we already touched on, is that transcription and translation occur simultaneously. Okay, again, prokaryote does not have a nucleus, so the ribosomes can essentially accept the mRNA as soon as the RNA polymerase is done pumping it out, or as it's pumping it out. Okay, and the 5th main one is, if you remember, the prokaryotic genome is arranged how? How is the prokaryotic genome arranged? It's arranged in a ring called a plasmid. And when we replicate the plasmid, we pretty much just start with 1 replication fork and we can keep going. Versus eukaryotic DNA, everything's wrapped around, the heterochromatin's wrapped around these histones. It's packaged up nice and tight. We have to go in and pull out certain segments that we want to replicate. So we're going to potentially have multiple replication forks, or we're going to have different replication forks in different areas. Versus the prokaryotic genome, we kind of want to keep certain genes together so that we don't have to go around the circle and replicate different genes as needed. So an example of this is an operon. And essentially what an operon is it's just a strip of RNA, or DNA, sorry, that has multiple genes on it. And what we call those is we call it a polycistronic segment. Polycistronic. And what that means is that when we transcribe our DNA in a prokaryote, it's going to have multiple genes on one RNA. Does that make sense? So again, a famous example of this that we're going to look at later is the Lac Operon. And so the next part we're going to jump into is how we actually regulate the transcription of genes in eukaryotic organisms. Okay, so there's three methods that we can use to regulate gene expression. One of them being is just regulating the access to the basic resources that are needed. So regulate access to basic resources. And that's things like DNA. We can regulate when DNA is transcriptionally active or not. We can also regulate when transcription factors are present. All the basic machinery that is needed to start transcription, we can regulate those elements to ensure that we're only producing genes when necessary. Next, we can also regulate the production of the product from these resources. And finally, we can regulate the lifetime or the lifespan of the product. And so essentially in this diagram here, we have our transcript being produced. It's going down, going through alternative splicing, getting the 5' cap, poly A tail added. Those are all post-transcriptional modifications. And the reason we add these two, just a little tangent, the reason we add this 5' cap and poly A tail is because when we exit the nucleus in eukaryotes, there's RNases that are in the cytoplasm that are immediately going to start degrading this mRNA. And so we need to protect the ends of our RNA, and the way we do that is by adding this 5' cap and poly A tail. But once this mRNA is out in the cytoplasm, it has two destinations. It can either be degraded, or it can go to the ribosome, be translated into protein. So again, these are three different ways we can regulate gene expression within eukaryotes. And as you guessed, there are more methods of gene regulation. But these are going to be more specific to covalent modifications and things like that of our DNA and the machinery that packages our DNA up. So at first we need to define a few terms. Heterochromatin. Heterochromatin, a way we can remember this, is it's the highly coiled DNA. Heterochromatin, highly coiled. Okay? And this is essentially our DNA that's wrapped around histones, which, if you don't remember, are proteins that form a nucleosome when the DNA wraps around them. And it's just a way for us to package up everything into a nice, neat package that is only going to be transcriptionally active when we want it to be. Euchromatin is the less condensed DNA. So this is when DNA is replicating and we've unpackaged our DNA. It exists as euchromatin. So there's a few things we can do to essentially regulate transcription at the DNA and histone level. So DNA methylation, and I want to preface this with, whenever you see the term methylation, it's generally going to mean to mute. So DNA methylation, when we methylate our DNA, we add a methyl group to DNA, it mutes transcription. Okay, so we get a decrease in activity. And that's true for most of methylation. Histone methylation is one of the weird ones that most of the time it mutes. Okay, we can also methylate the histone, which is the protein, again, that our DNA is wrapped around in heterochromatin, we can methylate that, and it'll also silence and reduce the amount of transcription that's occurring. One of the other two main mechanisms is histone acetylation and histone deacetylation. In histone acetylation, what we essentially do, and I have a slide on here, and I'll get into more of the mechanism of acetylation, but we add an acetyl group to our histones, and that essentially allows it to be transcriptionally active. So it's going to increase transcription. And on the contrary, deacetylation will then decrease transcription. And we'll look at these two mechanisms later on in the future slides. Another thing we can do with the heterochromatin itself, apart from covalently modifying it with acetyl groups or methyl groups, etc., we can actually use this molecule called sweet sniff. And what this does is it's basically an ATP-dependent process. So it's going to require energy, but what we do, almost like a fishing reel, we move these nucleosomes away from each other. And if you imagine what's going to happen in a fishing reel, when you start to pull on the strands or the reel, it's going to unravel. And so this DNA here is going to unravel, and it's going to give our transcriptional unit, transcriptional factors, everything, it's going to give it access to start transcribing those genes. Okay, so this is a different way of allowing transcription to take place by essentially moving the nucleosomes, moving these histone proteins away from each other, allowing certain segments of DNA to be exposed. And so again, these are just the covalent modifications of DNA and histones, where we can either methylate it, and that will again decrease transcription or silence the genes, or we can acetylate it. And I want to go a little bit more in depth into the mechanism of acetylation. So essentially what happens is we have an enzyme here called histone acetyltransferase. And what histone acetyltransferase is going to do is it's going to acetylate this lysine residue. And so in normal nucleosomes, normal histones, these side chain lysines are positively charged. And as you know, DNA is negatively charged because of the phosphate backbone. So these positively charged lysines and negatively charged DNA are going to attract. And so that's what aids in the packaging of the heterochromatin. But when we acetylate the lysine, it gets rid of that positive charge. And so what happens is the negative charge now has a reduced electrostatic attraction to the histone, so the DNA starts to come off of the histone. And that allows for an increase in transcription. Sorry for my handwriting, it's terrible. On the contrary, we can then go backwards and deacetylate this lysine to cause the heterochromatin to reform and to again decrease transcription. Recently there's been some studies and some therapeutic agents that have been developed that are called HDAC inhibitors. These are called HDAC inhibitors. Let me get rid of that. And so let's think about this for a second. If we have a histone deacetyl, I'm not even going to try to pronounce the enzyme. I forgot what the enzyme is, so whatever. If we have an inhibitor of this enzyme, let's think about what this normal mechanism is doing. If we deacetylate this lysine, we're going to cause heterochromatin to reform, and there's going to be a decrease in transcription. By inhibiting this, we allow transcription to occur more frequently. This drug would be useful in certain diseases where there's a underexpression of certain genes. Okay? So we can use an HDAC inhibitor to cause more expression of certain genes that are lacking in certain disease pathologies. So another mechanism of regulation is at the mRNA level itself. And it's a process known as mRNA turnover. So essentially, in this example here, we have an mRNA transcript that codes for a transferrin receptor. And what transferrin receptors do is they essentially allow for iron to come into the cell. Okay? So in times of high iron content within the cell, so if we have increased iron, we don't need to allow more iron in. So when this mRNA is produced, it's just going to get degraded. And that's what we see here. But what happens is when we have low iron, we actually get the activation of these little proteins here. And these are IREBPs. And what they essentially do is they come and they stabilize these segments of the mRNA to prevent the degradation of that mRNA strand. So by preventing the degradation of that strand, we allow that mRNA transcript to get translated, and we can now inject these receptors into the membrane, allowing more iron to come in. Okay, so essentially, we're going to turn this into a high iron environment by transcribing and translating this mRNA segment that gets stabilized by this protein here. Another mechanism of regulation of transcription is with this DNA binding protein. And this DNA binding protein is called the zinc finger. And essentially, the key component here is that it uses zinc to coordinate binding to DNA. So the zinc finger is going to recognize the major groove of the DNA. Sorry, that's the minor groove. The major groove of the DNA, and it's going to attach itself and also act as a regulator of transcription. And so last but not least, we're going to talk about the Lac Operon. And the Lac Operon is seen in prokaryotes, and it's also a method of gene regulation, but it introduces some new components here. So we have a promoter region, which is similar to the eukaryotic promoter region. It essentially is a place where RNA polymerase can bind and start to transcribe these genes. We also have an operator region, and this is where this protein molecule called the repressor binds. And when the repressor is bound, the RNA polymerase cannot work. It cannot go and transcribe these genes, so we don't get any production of these 3 Lac genes. And what these Lac genes do is they essentially break down lactose. Okay? So, in periods of starvation where we have no glucose, or these organisms, sorry, not us, these prokaryotes have no glucose, they're going to basically respond to lactose now. So when lactose is present, it can bind to this repressor, pop off the repressor, and now our RNA polymerase can transcribe these genes. Also, another point that's not shown here is when there's no glucose present, we get this molecule called cap cyclic AMP. And what cap cyclic AMP does is it helps to recruit RNA polymerase during periods of starvation. When there is no glucose present, cap cyclic AMP will also come to this promoter region and start to recruit RNA polymerase in tandem with the lactose binding to the repressor and popping it off the operator. And once this is all the machinery set up, we can transcribe these Lac genes, and they can basically be produced to break down lactose. That's all I have for today. I hope this video was extremely helpful, and please feel free to reach out with any questions that you may have.